Talk to me. Hey, 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 hey.
Hi, everyone, and welcome to Night School. I'm Lynn from the Nightlife Programming team.
I'm Christina, and I'm Lynn's co-host and also the event producer.
Thanks for spending your Thursday night with us. We are super excited for tonight's Night School. Tonight, we got Sloths, a full hour all about your favorite slow-moving mammal.
We actually are doing this because somebody suggested having a Night School about sloths. So if you're watching, I hope you are, and I hope this makes you happy. First up, we have Nestor Correa, who's joining us from Panama. He's the executive director of the Pan American Conservation Association, a wildlife rescue organization in Panama, and he'll talk about their work to rehabilitate orphan sloths. He also has about 4 baby sloths with him, and you'll get to meet them. We just saw them a few minutes ago. Next up, we have Bryson Voirin, who studies sleep in sloths. Do you think sloths sleep a lot because they're super slow? You're going to find out. And he's also going to talk to you about how to catch sloths, which actually isn't super easy. Next up, we have Dr. Emily Lindsay. She's an assistant curator at the Brea Tar Pits, and she's going to run you through, 50 million years of sloth history. And then finally, we have Emily Fountain, who's a research scientist at UW-Madison. Who's going to talk about why sloths are so okay with their fur covered in algae and ecto-parasites.
And as always, tonight's program is live, so say hi. Let us know where you're watching from. If tonight's your first time joining us, or if you've been with us the last 7 or 8 months, and share any comments or questions you have in the chat. We'll have Q&As after each of the presenters, so get all your sloth questions ready. Let's head off to Panama with Nestor.
I'm frozen solid, sorry. Hello, welcome to tonight's presentation. My name is Nestor Correa from the Pan American Conservation Association. We're streaming from the beautiful and warm Panama City. It's 10:06 Eastern Time, 82 degrees Fahrenheit with 90% humidity, perfect environment for sloth life. You will see that behind me, right around here, we have these. But later on, you will be able to see them closer instead of my face. Don't worry. It's a pleasure to be here. First, I would like to thank Lynn Kuhn and Christina Fon from the California Academy of Science for the invitation. Before we start talking about baby sloths, let me make a short presentation about us. Our organization dedicates to wild rescue and rehabilitation for the past 15 years. And during that time, we rescued thousands of wild animals with very small support from our government. But I will explain you more about that later. Species we rescue the most are sloths, anteaters, hawks, owls, opossums, squirrels, crocodiles, snakes, and different species of primates. APPC is leader in wild rescue, and we focus on sloth in particular. We rescue and care for sick, injured, and orphan wildlife. We provide them with the veterinarian treatment for the one who needs the treatment, and sometimes we also rescue animals that just need a few hours, 40 hours of observation before we release them back into the wild. We run a small visitor center, and we, of course, do environmental education to create awareness in the Panamanian community. We started back in 2005, and we so far relocate more than 5,500 animals back into the wild. Deforestation, it's our big challenge for us, and the biggest threat for sloths and other species of wildlife we have. So we dedicate all our effort to try to rescue and relocate animals that are in areas that used to be forests that are now fragmented to areas where they are going to be able to survive. Our rescue center is located in a beautiful area called the Soberania National Park. That is one of the national parks we have in the Panama Canal area. If you're familiarized with Panama, you will know that we have a canal that was built by the US government back in 1903 to 1914. It's a very rich biodiversity area. It's a great location for us because we can do rescue and rehabilitation and release animals back into the wild. That national park is connected to 3 other national parks and makes around 1 million hectares of rainforest, all in danger, sadly, because of urban expansion. We cooperate with the local eco police, with the Ministry of the Environment on the rescues. Basically, what we do is we work around the clock. We receive the animals, we do the treatment, and we try to return them back into the wild as fast as we can. We do not have a big staff. We only have 7, and we have volunteers that go from 3 to 6, depending on the needs we have. Everybody on our team have a big heart, and they love working with animals, and that's the main reason we can fulfill our mission. We have a group of very dedicated people, but sadly, you are not going to be able to meet them all. But some of them are in the pictures, and you will meet one very special member that is going to be moving around behind me, but I will tell you who is she later. Panama, like many other cities, is expanding, and forest is shrinking. We are creating more and more fragmentation on the forest. You probably know what do I mean with fragmentation. We're just building roads, highways, residential areas, shopping malls. Panama is happening exactly like in many other places, and that is putting animals more in danger and is increasing the roadkill. In Panama, people tend to say that we are being invaded by the animals are invading us, but it's actually the opposite. We are invading them, and sometimes people don't do research very well. They buy a new house, without knowing that that area was actually rainforest. And then they start calling us, complaining about snakes on their backyard, coatis attacking their cats, crocodiles. And, not everybody is like us. We enjoy wildlife. We love when we have the chance to see any animal moving around, but most of the people is going to be complaining, and sometimes we have to do a rescue that is really taking an animal from his natural habitat to a different location. And that causes problem because animals will get disoriented, and it's basically putting them on jeopardy. As you all know, sloths are the slowest mammal in the world, and some of my colleagues are going to be explaining more about the biology. I'm not going to be going into the biology of sloths too much. I will just give you some basic information that you want to know. But the main reason they're so slow is because of their diet and their slow metabolism. Because of that, they become the #1 rescue for us. And the main reason we need to rescue them is because they get disoriented, they get attacked by domestic animals like dogs, or they can also get electrocuted in the power lines. Now, let me tell you a short story before we jump into the babies, because I know you want to get close to them, close to the action. When animals get disoriented, it's like when you are trying to go to the supermarket. If you have highway roads, you are not going to get lost, or if you have a GPS, you will be. It's going to be easy for you to get to the location you wanna get. Animals is different. They get oriented using the forest. When we take the forest away, or we cut it in pieces, they don't know, and they just get. We rescue many animals that they just go frozen solid. They just stay there, stand still, looking around like, where do I go now? Especially with the sloths. And since, for example, Hoffman, the two-toed sloths, the ones we have right here, are primarily nocturnal, they will be moving in the night, and once the sunrise comes, they don't know where to go, and that's where we have to go rescue and do relocation. And we rescue every year more than 450 animals. More than 30% of those are sloths. That takes me to the action, the baby sloths. But before we jump into baby sloths, we are going to play a short video that we produced with a volunteer from London, Lucy Hollingston. She did this video for us. We use this video for fundraising. So Christina is going to play the video for you guys now. And it's at 1 minute 30-second video, and then we will be feeding baby sloths.
Hi, my name is Nestor Correa from the Pan American Conservation Association down in Panama. Our orphan sloths need your help. Every year, we receive hundreds of orphans and injured wild animals, with baby sloths representing the largest number. Almost all the babies we currently have lost their mothers as a result of habitat destruction. Sadly, as forests shrink, more and more sloths are wandering into urban areas where they become vulnerable to road traffic collision, domestic dog attacks, power line electrocutions, and even human cruelty. Our dedicated team of staff and volunteers works around the clock to provide the animals with food and expert veterinarian treatment.
Hi, my name is Chelsea Morton. I'm from Baltimore, Maryland. I began volunteering with the APPC in 2016, and I've been here ever since, all because I love these amazing animals.
Your donation will be used to buy food, medicine, veterinary supplies, and building materials for brand new enclosures.
So please give what you can and help us save Panama's orphan sloths. Thank you.
Okay, so we are back on the action. Behind me, we have my lovely wife, Giselle. She's actually doing some feeding. We will be doing some goat milk later. Now, a few very important things for us, because we like to credit who is actually helping us. Now, you may wonder, how do we manage to keep working for 15 years without government support? At local level, the Gamboa Rainforest Resort, it's an eco lodge inside the national park where we are, provide us with free housing and infrastructure. These facilities works perfect for us because we can actually use all the funding we raise for the animals. Throughout the years, we developed alliance with foundations from the United States. The first one was with our friends from SeaWorld Busch Garden Conservation Fund. They are led by our good friend Rob Jordy, who's a big fan of us. He likes to come and help us all the time. They provide us with technical and financial support since then, especially now in these difficult times with the global pandemic. Other partners include the Indianapolis Zoo, Miami Zoo, through the Ron McGill Foundation, and we also receive support from individual donors. So if you want to support us, just visit our website at www.appcpanama.org. Now, no more talking. Well, actually, I will keep talking, but you will be able to see the baby sloths. Let me see if we can get closer to them while I keep talking. So the babies we have right here are just a few months old, and they are with us at the house. Yes, we are at the house, or well, we are with them. Babies will be eating every 3 to 4 hours from 8 a.m. to 9 p.m. So it's a lot of work, between 15 to 35 milliliters of gold milk formula. We use a special brand called Mayenberg because comes in powder, and that helps them because they don't really tolerate the lactose and the fat very well. When babies are more than 2 months, we start introducing leaves, and after 6 months, will just be that. Sloths are slow, but they can move fast, and when they need it. Sometimes some people call them the fast and the furious because they will be fast and aggressive. It's not that easy to handle them, especially the Hoffman two-toed sloths. They have a very strong grip. They will be protecting themselves, and they will bite you. They have 18 teeth, and 4 of those are actually canines. They're arboreal mammals, so they spend most of their life at 40 meters in the canopy, hanging upside down 90% of the time. They can live more than 20 years in the wild. In Panama, we have 3 to 6 species, 3 of the 6 species. We have the Hoffman, we have the brown trout three-toed sloth, and we have the Pygmy sloth. They are related to the anteaters and the armadillos in a superorder called the Sonartrans with an X. That means special articulations on the lumbar area. They are very, very old. Their ancestors were called the ground sloths, and they were up to 18 feet tall, standing in a vertical position, weighing like a ton, like an 4 tons, like an African elephant. They digest their food in 2 weeks, and they will be going to the bathroom every 3 to 7 days. Their main predators are the harpy eagles. We don't have too many, sadly, for the environment, but good for the sloths. This is a big raptor that weighed up to 25 pounds with an expansion of wing of 2 meters, and the claws are stronger than they're so strong they are compared with grizzly bear paws. We may think it's easy to be a sloth, but it's not. In fact, it's very difficult since they have to move around to find their food, and that's when they become vulnerable to the predator. So let me see if I can get closer. There we go. Here we go. Let's see. Okay. Now, since they need to move around, I'm sorry, I'm lying on the floor so I can read this for you guys. When they become vulnerable to the predators, the other problem we have, it's not only that, it's actually road, power lines, expansion of the urban areas. Those are actually putting on risk. Once the sloths are more than 6 months, we transfer them to our rescue center in Gamboa so they can start their journey back into the wild. This takes 9 to 12 months. And when sloths are around 1.5 years old is when we place them in the outdoor habitat that we built a couple of years ago. And then they spend 3 to 6 months there before we start getting them ready for release. What we do is we use tracking devices. We use telemetric system that is with antennas, and that they have a range of 50 square kilometers. But since we are in the forest, it's very difficult for us to track them with those devices. GPS is much better, but it still is useful. Our main goal is to release them back into the wild. Sadly, sometimes some sloths are not able to go back into the wild because of their condition. They get sometimes too used to be with humans, or they're disabled. Okay, before we finish the presentation, I will ask Christina to play the last video. That it's a 1-minute video of Chelsea Morton. That it's our researcher on the forest, and she's the one leading the project to release sloths back into the wild.
Sloths are one of the most common wildlife species rescued by the Pan American Conservation Association here in Panama. When sloths arrive as orphans, they are hand-raised in captivity until considered suitable for release back into the wild. Here we are at the APPC soft release enclosure. This is the soft release enclosure, where sloths will spend 3 months before being released back into the wild. Before being placed into the enclosure, each sloth is fitted with their own VHF tracking device, which will allow for post-release monitoring as we watch their transition from captivity to life in the wild.
Okay, guys, thank you for watching the presentation. Thanks again to Lynn and Christina for the opportunity. And remember, if you want to support APPC, go to our website, appcpanama.org, and I will just try to get you as close as I can to the babies.
Hi, Nestor. I think we have time for a couple questions.
Okay, I'm ready. Let me go back here.
Cool. So first one, can you tell us a little bit more about the baby sloths that you have behind you? What are their names?
Oh, okay, that's my wife's department. Let me ask. Gis, ¿nombres de los perezosos? No tenemos nombre.
No, es Sara.
Sara?
La que está conmigo.
This is, let me see. This is
Sara.
Sara. Uh-huh.
Gul.
¿Cómo?
Gul.
Gul. Uh-huh. Skype. Okay. We're running out of names because we have so many. Uh-huh. And? Goose.
Oh my God.
Goose. Goose. Okay. Well, we have one without name, so we can actually raffle that. You can, the person that asked, we can give her the opportunity to name the number 4.
Yeah, viewers, this is your opportunity to name a sloth, so throw in some suggestions. So our second question is, what is a sloth's diet? What are the flowers that they were eating in the background?
Okay, in the forest, they will be eating a big variety of plants. There's one that we call the guarumo, that it's a cecropia. It's a very pioneer tree that we have everywhere. They can also eat mango leaves. They eat gumbo limbo, that it's actually a tree you can actually find in the U.S. and South Florida. And there's more than 85 different species. But the most important thing to know is that the sloth will climb to the top of the canopy and they will be eating just the very, very new leaves. Now, the flower that we feed them is the hibiscus. They love flowers. Flowers are soft, easy to chew. Sloths don't really chew too much. They basically take chunks and they swallow. Also, in captivity, we gave them, when they're babies, spinach, carrots, and bok choy.
Nice, healthy. And we have time for one more question. Do you release animals back into the wild? And if so, is it a problem that they are now used to humans? How old are they when they're released?
Yeah, it's very difficult. It's a very difficult challenge. I'm not going to lie to you. We released 12 slots in the wild in the last year. We lost a few already. Some are actually doing fine, but the biggest problem is being not actually getting used to humans. It's actually dealing with the predators and looking for food. The biggest problem we have with we're talking about specifically about sloth, because we can talk about different species, but it's always difficult when you hand raise an animal. Sometimes it's basically impossible to release. Well, you can release the animal, but the animal's not going to survive. But if you want to increase the chances for the animal to survive, you need to try to separate the animal from you when the animal's still young. In the case of sloths, we try to isolate them before they reach the year. And if you keep them after a year, it's going to be very difficult to release back into the wild.
Great. Thank you so much, Nestor. I think that's all the time we have for questions. Thanks for joining us tonight.
Thank you. Bye-bye. Take care. God bless.
Thanks.
My name is Bryson Voirin, and I'm a sloth neurobiologist. I spent my PhD work studying sleep in sloths in the Panamanian rainforest. And as you can see here, here is a three-toed sloth named Chispa with a nice little hat on it, which records its sleep. Now, a little bit of background about sleep research. Most people that I work with are medical doctors, and they study sleep in humans. And the basic question of all sleep scientists is, why do we sleep? I personally love to sleep. I get tired all the time, and I can pretty much always take a nap. And the biggest question is, why do we sleep? It's a period where we can't eat, we can't mate, we can't work, we can't really do anything. We're actually vulnerable. And so everything ever studied has been shown to sleep in some capacity, but we don't really know why. And this is a great picture from Salvador Dali showing that even giant, scary flying faces sleep. And it shows the fragility of sleep. So it's posted up on these little posts. And if you hit one of them out, the thing would wake up. And so sleep is different than being unconscious or dead because it's rapidly reversible. Now, whenever I give talks, I always try to ask people, how do you define sleep? You know, sleep in a sloth or sleep in a person, what does it really mean? You know, if someone's dead, they're not asleep, or if they're unconscious, it's not asleep. So as sleep scientists, we have developed a couple of criteria to describe what sleep is. It's a period of sustained quiescence, of specific posture for the individual or the species. So a cat will sleep curled up, a human will sleep prostrate in bed, a sloth will sleep in a tree just doing nothing, fish will float. And so every species has a specific posture. It's also very easily reversible. So if I could poke you with a knife or a stick or tickle your feet, you're going to wake up real quick. Even a sloth, you go like that, it'll wake right back up. And it's also homeostatically regulated, meaning that if you don't sleep for a long period of time, you'll make up that sleep debt over the next few nights. And it also, you're not as prone to external stimuli. So if someone walks by your room, you might not wake up. And kind of the purpose of what my research has been is looking at the comparative way to look at sleep. So you could study sleep in humans your whole career, but I actually like to take sort of the holistic approach and look at how different animals throughout the animal kingdom sleep in different ways. So blue whales and fruit flies sleep in some ways in the same ways, but in some ways very different ways. And so I like looking at the diversity of sleep to see what are the commonalities throughout the animal kingdom and what are the differences throughout the animal kingdom. And I love this picture by the scientists from the 70s looking at all these animals sleeping together, forming a human brain. It's a little psychedelic. And another question is, how do we actually study sleep? So you can stare at a sleeping person and kind of figure out if they're asleep or awake, but that's not actually the easiest way to study sleep. Behaviors can be very deceiving. Dolphins can sleep with half of their brain. Ostriches actually sit with their head up, with their eyes open, and they're in slow wave sleep. Cows can chew and like masticate when they're actually in slow wave sleep. And so it's not always like the easiest way to just look at animals and say, okay, they're sleeping or not. And so we actually have to look at the brain behavior, so the neurological type of sleep, and that's a little bit harder to do. We have to use what's called an EEG, which I'm sure most of you probably heard of. Maybe you've had them if you have sleep apnea. It's an electroencephalogram. Basically, it looks at the changing brainwave patterns going throughout your cortex. And so using these very sensitive, basically voltage meters, we can actually detect the changes in the brain. Because when you're asleep, actually, your brain is on off. It's just firing in different ways. And so slow wave sleep, if you look at a graph of an EEG, it's these large slow waves going up and down. REM sleep, or rapid eye movement sleep, is when your eyes are moving, you're dreaming, and your brain is actually highly activated. It's called paradoxical sleep as well, because your brain is just as active when you're asleep as if you're awake. And the lab that I worked at kind of pioneered the research of wild sleep recordings, which sounds kind of funny to talk about, with sloths. If you look at what we knew up until a few years ago, every animal that had ever been studied was studied in a cage or in a zoo or in a box in a lab or something like that. And it's a very artificial way to study sleep in a natural organism. If I were to study sleep in humans in transatlantic flights, my entire career, never once actually study sleep in a person in their bed at home, I would have a very unrealistic or probably biased view of the way that humans sleep. And so what we thought was, well, we think we know how all these animals sleep in the animal kingdom. We've studied them since the 70s, but I bet you it's all BS. You know, when you're sleeping in a box versus at your home or in the wild, you know, you're not foraging, you're socially isolated, you're probably scared, you're being deprived of the sights and sounds and smells of the jungle. You don't have predators to worry about if you're a sloth, harpy eagles and jaguars and ocelots and pumas to kill you. And also, there's a growing body of research looking at the brain chemistry changes of an animal in captivity. A lot of people really don't like zoos, and I understand that to some degree. And actually, if you look at the brain chemistry in animals like a restless tiger, they actually are depressed. And so there's a lot of evidence showing that if you look at an organism in captivity and an organism in the wild, it's almost like a completely different species. So when we first told sleep researchers we want to study sleep in sloths in the wild, they thought we were absolutely crazy. So we showed them this kind of funny diagram, and they were like, all right, whatever. We got some crazy Russian scientists to create a neurologger that is 1 square centimeter in size and about 1 gram in weight, and it records EEGs for 2 weeks. So it was a very cool thing, because usually EEGs are this huge computer with stuff all over your head, and it's connected to computers. Doesn't really work well in the rainforest. So once we got these little loggers, I set off to my natural habitat, the rainforest, and realized that, well, actually catching sloths, how in the hell do you do it? Sloths are usually in the very top of the tree in very small branches. I can climb quite well, but I can't be on a branch that's 110 feet up, halfway up the tree, that's this big around, because they would snap and I'd die. And so we had to devise a certain technique to catch them. It's actually pretty tricky. So we think of sloths as these like nice, cuddly, happy creatures. You can just, you know, call them and they're gonna come to you and be really sweet. In actuality, sloths can be really dangerous. I actually had to go to the hospital by getting bit by one. Actually, not this one, but one just like it, a Tuto sloth. You think they're really slow, but they're not. But they actually are really slow. The way I actually catch them, though, is I have to climb up using jumars, ascenders, harnesses, a huge slingshot to shoot a rope up, and climbing ropes. And it seems like, you know, as a human, I should be able to catch these really slow mammals. They don't move much, so as long as I can figure out where they are in the tree, get up there. 9 times out of 10, I got outrun by them, because for me to set all my crap up in the tree usually takes about 30 minutes. I have to shoot a slingshot up, and then when the rope comes down, tie a bigger rope onto it, pull it over there, climb up there. And sloths do move. They don't just sit there. And so even if I'm climbing as fast as I can, a sloth moving very slowly across the branches can probably get 30 or 40 feet away. And my arms are only so long, so I can't reach and grab them. So I have to come back down, get laughed at by all my assistants and friends because I got outrun by a sloth again, and reset and do it again. So there's been days where I've spent half of the day trying to catch one sloth, and then finally gave up in disgust and went to the bar because I couldn't catch a damn sloth. So once I am able to get close enough, I use a snare pole and I grab one of their arms, and then generally they'll reach around to the snare pole, grab onto it with their arm, and then I can rappel down quite nicely and bring it to the ground then. So a little bit about the research findings that I had, which is always, as a scientist, I love to talk about on my data. The 3-toed sloths that we studied was the first ever wild sleep recordings. In the wild, we found they slept about 9.5, 9.6 hours every 24 hours. In captivity, they slept 15.6 hours, which is a significant difference. I presented at a conference, and one of the scientists was like, yeah, but how big was the cage you had them in? They just didn't fundamentally believe that we did this in the wild. And I was like, well, it's in sloths in the rainforest, and we showed this huge difference. So there's a lot of possibilities of why there's this big difference. There's a big train of thought about predation pressure driving sleep architecture throughout evolution. If you're a sloth, basically, you're living in the tree, you're vulnerable. And so you want to sleep at opposite times of your predators. And so it's possible that they sleep at very specific times so that they're hidden, they don't move, they're not eating at all. at dawn and dusk because that's the time that most of the predators in the rainforest are active. I also did some studies on Hoffman's two-toed sloths, which are the aggressive ones, which the reason I went to the hospital was I was caught one for the first time, forgot that they turn their heads about 200 degrees around, and it bit me in the hand. And they do have little fangs, and it went into my finger, and it wouldn't let go. So I had to pry its mouth off there, and then went to the hospital in Panama. And the Smithsonian called ahead to tell them, hey, there's stupid American guys coming in there, you got bit by a sloth. Surgeon meets me laughing hysterically like a witch, doing like, what is wrong with you? You got bit by a sloth, were you asleep? And the whole hospital's laughing at me, kind of antagonizing and making fun of me. And I just had to laugh too, because it was so ridiculous. And they taped up my hand in a big pink bandage. And basically I'm infamous anytime I got to Panama. Everyone's like, wait, you're the guy that got bit by a sloth, right? And yes, I am. So I was bitten by a sloth. This actually happened twice now, but the other one just kind of nipped me a little bit, but that one was pretty bad. So this guy is the one that can actually bite you. The two-toed sloths, when they studied them in captivity, they slept about 22 hours, but they weren't really sure because no one ever did neurological or EEG sleep recordings on them. They'd only looked at them and said, okay, it's not moving, it's moving for 24 hours at a time. We found that they're interactive about 16 hours a day, and they slept in EEG-based sleep for about 11 hours. So it's a big difference than we thought. So in actuality, these guys aren't as lazy as you think, or maybe they're actually more lazy than you think, because they're only sleeping 11 hours, but the rest of the time they're just sitting there doing absolutely nothing with their eyes closed. So I'm not really sure. It could go both ways if they're more lazy than we thought or less lazy than we thought. They could be sitting there very quietly doing higher level mathematics and calculus in their mind, thinking about multidimensional hypothesis and the central consciousness, but probably they're just thinking about leaves. My favorite sloth species, which I don't know if you've heard of it or not, is the pygmy sloth. Sloths are really cute to begin with, but these guys are about 2/3 the size of a normal sloth. So they're very cute size, and they're very sweet. They only live in this one island in Panama called Escudo de Veraguas, and they only eat mangrove leaves, which is very unusual. Most three-toed sloth species, they eat like 27 to 35 different species of leaves from trees and lianas. These guys eat one, which is really weird. They're also quite a bit smaller. They're a little bit slower than normal, which is impressive. And their behavior is different. They're much more tame. So sloths on the mainland Panama, they'll actually run away from you. They'll hiss at you a little bit. These guys just kind of sit there and look at you and they're like, what's up? And what we found is when we did the EEG recordings of them, some interesting things. We found they slept about the same as other three-toed sloths in the mainland Panama. They slept at different times, so it could reflect the fact there's no natural predators there, which is pretty cool. But the most interesting thing is when we looked at the, it's called sleep spindles. So in the slow wave area, if you think of a honeycomb spindle, that's a hallmark of mammalian slow wave sleep. The spindles were much bigger than you see on other three-toed sloths. And we actually checked with a drug specialist because it reminded me of something I learned in grad school. And I was like, wait, this is benzodiazepine addiction. And it turns out that is a hallmark of benzodiazepine addiction are these grossly increased or greatly increased spindles and also certain densities in the EEG. And we talked to some specialists on mangrove leaves, and they said actually there are certain endophytic funguses that have naturally occurring different types of benzodiazepines in them. So like Valium, basically. So it's possible that these guys are just addicted to mangrove leaves on this one island because they have benzodiazepines in them. So that would actually explain why they're more chill than normal. And we actually tried a little bit. We put them on the edge of the mangroves, and they would just basically very slowly run back into the mangroves to get more of them. So it's a very bizarre thing, which I would love to study more when we can travel again. Kind of a funny thing. And the neatest thing about pygmy sloths is they actually swim in the water. They're better swimmers than they are actually they're better, break there. They're actually better at swimming than they're at walking, which is kind of cool. If you wait there for several days at a time, you'll actually see them swimming around. So I filmed a bunch of documentaries with the BBC about these, and we spent 2 months waiting for these guys to go in the water to cross between mangrove patches. So it requires a lot of patience to study sloths. I'm sure you guys got lots of sloth questions. Hopefully I'll be able to answer any of those if I have service driving to Utah to go not look for sloths, but to go hang out. I'd love to tell you some cool sloth facts, but I'm sure throughout the night you'll probably hear other people telling cool sloth facts. My favorite one is all sloths have 3 toes. It's the number of fingers that differs. So when people talk about the two-toed sloth, the three-toed sloth, they all have 3 toes. It's just the number of fingers that differs. And my nickname in Spanish is the 5-toed sloth. Perezoso de 5 dedos. Thank you guys for coming and have a great night.
Hi. So as you guys heard, Bryson is actually on the road to Utah, so he's not actually able to join us for Q&A. We were hoping to have him live, but he's driving and that's just not safe. We did, however, get to talk to Nestor, and thank you for all the suggestions. He said he will name the baby sloth Mango. Up next, we have Emily Lindsay, who will talk to you all more about extinct giant sloths.
Hi, good evening, everyone. I hope you're all doing well and staying safe at home. So I'm Emily Lindsay. I'm a curator down at Brea Tar Pits in Los Angeles, and I study animals from the time period that site covers, which is the Ice Age, which ended about 11,000 years ago. And I'm going to be talking about some sloths that you may not be as familiar with as the cute modern ones we have today. So from all of the talks today, you're going to be hearing a lot about these two adorable creatures, the two-toed and three-toed sloths. And this is a way we show the relationships between creatures. So we know these are the only two living sloths. Obviously, they're more closely related they are to one another than to any other living creature. They're the only sloths we've got. However, some people may be surprised to learn that the Colepus sloths are actually much more closely related to giant sloths the size of hippopotamuses that don't live here anymore than they are to the bradypus sloths that tend to resemble them and have similar lifestyles to them today. So to put this in context, I'm going to give you a brief overview of the long evolutionary history of sloths in just a few short minutes. So this is how paleontologists like me think about time in the millions of years. So on the bottom of this screen, you have the time scale going back to about 70 million years ago. And at the top are the names of different time periods that we've divided those last 70 million years into: the Maastrichtian, which is the very end of the Cretaceous period, and then the time periods that make up the Cenozoic, or what we call the Age of Mammals. So just to put us in place, about 66 million years ago is when the large dinosaurs and about 75% of everything else on Earth went extinct in the context of sort of long-term processes of volcanism and climate change, and a very short-term process of a meteor smashing into the globe. This, of course, paved the way for the rise of mammals, as we talk about. So opening up all of this ecological space that was previously occupied by these large dinosaurs, and along with a changing climate, enabled a different group of animals to start to diversify and evolve and become abundant and dominant on Earth, and those were the mammals. And not long after this time, by about 62 million years ago or so, we actually see the origin of one of these groups, which is the Xenarthra. It's a superorder, so it's several orders together that includes the modern day sloths and their relatives, the armadillos and anteaters. Now, this group arose in South America. At this point, South America had just recently separated from Africa. Had previously also been connected to Antarctica and Australia and Madagascar and India, but by this point it was completely isolated, and it remained isolated for about 60 million years, which enabled the origin and diversification of a lot of very strange creatures that lived completely in isolation for tens of millions of years. And one of these was the Xenarthra. So about this time, we also see the divergence of the one group of the Xenarthra, the cingulates, which are modern armadillos and their extinct giant relatives. A bit later than this, about a little over 50 million years ago, we see the divergence of the other two orders in Xenarthra, the anteaters and the modern sloths. And fast forward to this, so these groups, we don't have necessarily a great fossil record of all of these tens of millions of years, but definitely by the beginning of the Miocene, so about a little over 20 million years ago, we see a pretty rapid diversification, or large diversification in the ground sloth lineages. And this is based both on molecular data that we have as well as the fossil record. And so we have a pretty remarkable suite of creatures living in South America and eventually making their way up into North America and the Caribbean, right up until almost the present, when, again, about a little over 10,000 years ago, the vast majority of those creatures disappear. And that brings us to the relatively depauperate fauna that we have today. So what do we know about these ancient sloths that used to be sort of some of the dominant creatures in South America for many millions of years? Well, we know they were actually an incredibly diverse group. This one up here is the one on top. The upper left is a sloth from the Yukon. They occupied a variety of areas from, the Arctic down through the tropics and down into Patagonia. There were sloths, you can see on the lower left, they got to be about the size of elephants. There were giant sloths that, when they got to the islands in the Caribbean, actually got small again. These you see on the lower right, we refer to them as the dwarf giant sloths. This is similar to what happened with the mammoths that made it out to the Channel Islands in California. They got out to the Channel Islands, and then because of this trend on islands where small things tend to get bigger and big things tend to get smaller, in order to make use of the limited resources, we ended up with mini mammoths on the Channel Islands. In the same way, we ended up with dwarf giant sloths in the Caribbean. And then in the upper right is, I think, the most interesting one. There were even sloths that adapted to have a marine lifestyle. They elongated their limbs. They developed very dense limbs, something that's known as pachyastosis, where their bones became denser to help with the buoyancy in water and help them be able to dive down. And we've demonstrated through looking at chemicals in their bones and teeth that they were, in fact, grazing on seagrasses and plants that were growing in the ocean. So a remarkable group of creatures that were able to develop very distinct lifestyles and occupy very distinct habitats over their tens of millions of years of evolution. We also have some clues into what these animals ate. These come from some typical sources. Obviously, we can look at the morphology, the shape of their skulls and their teeth. And ascertain that, like their modern relatives, they were vegetarians. We also are able to look at chemical signatures in the teeth and in the bones. So this image on top is a tooth that scientists did what's called serial sampling. So sloths have teeth that grow continuously, and so they're able to sample along the tooth and look at what it was eating during different periods. And from this study, scientists were able to determine that this particular one is from Belize, I think, and they found that there was seasonality at the time, and so the sloths were eating different things at different times of year. And then another proxy we have for diet is this image at the bottom, which is we actually have a remarkable record in both North and South America of sloth coprolates, or fossil poop. So these are found in caves predominantly, both in Patagonia and in the American Southwest. And from these, we've been able to look and actually identify the macrofossils of plants that were found in poop. They've identified from one group of sloths called Nothrotheriops that they were eating up to 70 or 80 different types of plants. And people have also looked at, done chemical analyses of the coprolites and been able to get information about the plants as well as DNA of the animals themselves out of this. So this is, it's a very rare record to have in the fossil record, to have original coprolite material. And so it's a real bonus. There are also places, again, these sort of high elevation dry caves where we've found actually preserved skin and fur and keratin from the claws of giant ground sloths. And so because certain members of these groups like to occupy caves and lived in, they occupied such a wide variety of environments, including some that are more conducive to preservation. We have a pretty good fossil record of them, which is really lucky for us. We've also been able to learn some things about the behavior of these animals. So there's a number of sites. The big picture here is showing an asphaltic site, a tar pit. It's not the Brea Tar Pits. It's a tar pit on the coast of Ecuador, actually, that I excavated. That was a bone bed comprising the remains of at least more than 20 giant ground sloths, everything from tiny little babies, maybe even fetuses, up through old adults. And based on the data from this site, which included not only the bones of these animals, but a lot of plant material that had been cut to a very specific length, which we determined were either the gut contents or coprolites, fossil feces of these animals, that they probably were inhabiting a marshy area. There was a group of them that likely all died together or within a short span of time. And we concluded that, at least for this particular species, in this case, it was the giant ground sloth, Erumatherium, which is a late Pleistocene sloth, along with Megatherium, one of the biggest sloths that ever lived. These were ones that got to be the size of male African elephants, were actually did inhabit social groups. Now, later this same year, another study on a completely different group of ground sloths called Lestodon, which were found largely in Argentina and Uruguay area. There was another site described that also purported sociality in this other group of sloths. So it's not unusual to find these bone beds with dense concentrations of sloth remains. And so that's something that's been able to give us insight both into the potential behavior of these animals, that they may have lived together in groups, which is very different, of course, from their modern relatives, as well as some understanding of the demographics and growth and ontogeny of these species, which is really exciting. Here's something else that's exciting that we know about sloths. We know that they overlapped with humans in at least certain areas. So the bottom two graphs are from research that colleagues and I have conducted in South America for a number of years, looking at radiocarbon dates, a way of measuring the age of fossils of different megafauna species. We have two represented here. One's from the Ultima Esperanza region in Patagonia. One's from the Pampas region in Argentina and Uruguay and southern Brazil. And each of those individual dots in the graphs represents a radiocarbon date on a specimen of an individual extinct large mammal. The bell curves, the colored bell curves that are, and gray boxes that are above the accumulations of points in these two graphs are the estimated dates of extinction based on the fossil record, or the estimated dates of when these animals disappeared from that region. And then on the far right on both graphs is a representation of the human fossil record in that region. And what we can see is that in both of these areas, in Patagonia, down in the very southern part of South America, and in the central eastern part in the Pampas, there were many thousands of years when both humans and giant ground sloths were occupying these areas. Now, if that wasn't enough evidence, we also published a paper last year. This is shown in the upper right. That's a rib of a giant ground sloth, a Megatherium, that has pretty clearly been butchered by humans. It was found in an archaeological context. We have lithic artifacts from there, and we have cut marks on the bone. So it looks like not only did humans coexist with these animals, but at least at some points they also ate them. So finally, what happened to the giant sloths? Now, these are animals that lived for tens of millions of years, quite successfully, diversified, spread to other continents. Why aren't they around today? Well, at the very end of the Ice Age, as many of you probably know, there was a huge extinction event. The silhouettes of animals that are shown at the top of this slide are representing groups of animals that were alive in North America, South America, Eurasia, and Australia at the end of the last ice age. And the green silhouettes at the bottom of those groupings are the only ones that survived to today. So there was a huge disappearance event of large-bodied mammals, very size-selective, very disproportionate. And unfortunately, like many other groups, the giant versions of our sloth friends did not make it. Only the smallest ones survived. This is the causes of this are still under debate by scientists. There were two things happening at this time. First of all, we were coming out of the last ice age, so we were undergoing major climate fluctuations. We have good evidence of changes in vegetation in different parts of the world in response to these fluctuations. And in some areas, we have good evidence that these vegetation changes directly impacted the megafauna populations and led to the extirpation or disappearance of groups of large animals from specific regions. At the same time, this is the point when humans are spreading to other continents around the world and rapidly increasing in population. And again, we do have evidence of humans hunting not only giant ground sloths, but other members of these large mammal groups. And so, you know, there's been this debate raging sort of in paleontology and archaeology for many years about which one of these things led to this large-scale extinction event. My feeling is probably it was a combination of both. You have climate changes that are clearly impacting megafauna populations, and then you have a novel, very effective predator arriving on the scene with very successful hunting technology and the social structure and cultural ability to transmit this technology between groups and coordinate between groups. And so probably there was an intersection of pressures that led to the disappearance of the sloths and many, many other large mammals in the world. So with that, I'd like to open it up for questions, and thank you very much for your time.
Hi, Emily. Thank you so much.
Hello.
For sharing. I think you touched on it briefly, but when we were kind of researching for this show, we read a bunch of really cool articles about the giant sloth boneyard you found, that was found in Ecuador. So I really suggest everyone Google that and learn more because it's really interesting. But yeah, we have a bunch of questions. So first up, in the diversity of ancient sloths, are there some that appear to be more grazers, or are they pretty much all browsers, just specialized to different food sources?
That's a good question. So there is evidence of grazing among some sloths. I believe in the myelodontid family, there are some that show evidence of grazing. And I don't remember if this is based on the isotopic signatures, so the chemical signatures in the teeth and bones, or another way that we infer diet is looking at the actual wear patterns on the teeth. And so if you're grazing, you tend to get more sort of scratches on your teeth, whereas if you're browsing, you get kind of more pits in the teeth. These are visible sort of under very high magnification. And so there are colleagues I have that work on these questions. But yes, there are some instances of grazing, but it does seem to be predominantly browsers.
Great, interesting. You work at the Brea Tar Pits. Were there ever giant sloths in California?
Yes, and in fact, one of my first slides, one of those, not sure how to get back to it. Oh, here we go. I don't know if people can still see my slides or not, but anyway, I did show an image that was from the Brea Tar Pits. So, oh, there we go. Okay.
Yeah, we can show it.
So this one, where I show the relationships with Cholepus, the image on the bottom is a Paramylodon harlani ground sloth from the Brea Tar Pits. You can always tell Brea Tar Pits fossils because they're dark brown, which is not the normal color for fossils. Usually fossils from this time period tend to be more white or gray, or occasionally may have a tinge of green or something from the sediments, but these dark brown, we call it La Brea brown. Those are telltale signs that they came from a tar pit. So yeah, we actually have a lot of sloths at the tar pits, mostly Paramylodon, we do have a little bit of Nothrotheriops, which is that's the Shasta ground sloth. So they're often found at higher elevations. They're not as common at the tar pits. They were a little bit slender. Oh, and somebody was asking about diet. So we think that, although I don't know that this has ever actually been demonstrated in a scientific study, they have these very long sort of gracile arms. And it's thought that they may have been actually like digging into ant and termite nests and getting some of their nutrition from eating these insects, which, you know, I mean, they're related to anteaters, so it wouldn't be, and armadillos, both of which eat insects, so it wouldn't be a great surprise to find that some of the sloths also adapted to that.
Huh.
Oh, and there's something else I forgot to mention that's crazy about sloths. So Paramylodon harlani and other mylodontid sloths, so Mylodon, Paramylodon glossitherium, they all have armor embedded in their skin. So this is a very unusual thing. So I mean, they're related to armadillos, which are armored, of course, and then the extinct relatives, the pompathirs, which are basically giant armadillos, and the glyptodons, which are just amazing. If you've never seen a glyptodon, you should Google it. They're like little Volkswagen Beetle animals. They're the ankylosaurs of the mammals. But sloths, the mylodontid sloths, and these are found from like Patagonia up to the Yukon, right? This group of sloths, they have these little pebble-like bones embedded in their skin. And you can see it like when we find fossilized or preserved mummified skin, like at Cueva de Milodon in Chile, you can x-ray it and you can actually see how all these little bones are put together in the skin. And you find them in sites, and it's very easy to mistake them for a rock or something. But if you know what you're looking for, you can actually find thousands and thousands of these little bones, osteoderms.
Wow. And does any of that still exist in sloths today, or has it? Or any evidence of that still exist?
Not that I know of. I've actually I don't work much with modern sloths. I've never heard of one with any of this kind of vestigial armor. And it's not in all the groups. It's only in the Mylodontid group family, and I think not even all of those. So that's something that I know colleagues of mine have been researching, is why does this one group have this very unusual feature? I mean, it's not a normal thing for mammals to have like dense armor inside their skin.
Yeah.
So how did that happen?
That's fascinating. And then finally, a lot of people wanted to know, do you know if the giant sloths were also super slow?
It's a good question. So giant sloths would not have been as slow as the modern sloths. Like they were big and they walked around and they grazed, but they were really awkward. Like most of them walk on like the sort of inside part of their hands and feet. Most of them could stand up on their hind legs. Certainly the biggest ones, you find their thigh bones are like this big. It's crazy. There's one in my last slide here. Which I guess
I'll add it
if you want to go back. But the last slide, what I'm sitting next to there is a tibia and femur. And so the really, really wide one there, that's the thigh bone of a giant ground sloth there, and of an Erymatherium. So, and it's showing the diagram actually shows it standing up. So we know that they could stand up. I don't know if they really walked much on two legs other than to sort of threaten things or just get over to the next tree. But they weren't like runners and jumpers, that's for sure. But they also weren't like completely incapacitated on land like modern sloths.
Well, thank you so much. That's all we have time for right now. But thanks so much for joining us, and there's lots of questions about this. So
thanks a lot. Well, feel free to send me an email or something. I'm very Googleable. So if anyone has other sloth questions, I love talking about sloths. So thank you so much for having me. This was really fun.
Yeah. Okay, and up next we have Emily Fountain, and she will be talking about sloths and their little ectoparasitic friends. So here's Emily.
Hi, I'm Emily Fountain, and I'm a scientist at University of Wisconsin-Madison. And I work in both the Department of Forest and Wildlife Ecology and also at the Wisconsin National Primate Center. Today, I want to share with you some of our work we've been doing on coevolution in sloths, and also some of the more recent stuff where we have been looking at some of the many ecto-parasites that actually live on a sloth. Before I get too far into my talk, I really want to acknowledge the people who have helped us with this project, because without them, we would not have been able to do any of this work. In particular, our field workers in Costa Rica, led by Giovanni Herrera, my colleague Jorge Mendoza, the IANO Foundation in Colombia, and this work was funded by Systemax Association and NSF. So coevolution is not a new thing. In The Origin of Species, Charles Darwin actually postulated about what was going on with a bee and a flower and why were they able to adapt so easily to each other, and what seemed like almost simultaneously change their behaviors to benefit one another. In 1980, Janzing came with a more official type of definition, which really the basics of coevolution is just a reciprocal evolutionary change between two organisms that are always interacting with each other and in the presence of each other. So how do we study coevolution? Well, one approach is to use genetics, which is the approach that my lab uses. And we use a process that's called phylogenetics. And what that means is we build phylogenetic trees of the species that we're interested in, and we lay them over top of each other and test for different types of coevolutionary events. So when I'm talking about a phylogenetic tree, you can see by the diagram down at the bottom, these are different phylogenetic trees that are being represented. And we can kind of go back to the case of Darwin with his bees and flowers and look at them as represented by that. So the phylogenetic tree that is clear with the black outlines, we can think of this as flowers. At the very base of that tree would be a common ancestor that would be shared by these flowers that we're looking at. And after a certain period in evolutionary time, they can diverge from each other, which are the different branches that are coming off of the tree. And then at the very tips of that tree, we would see these species, and those would be the species that we're interested in. So in this case, with the clear one with the black lines, it would be different flower species or families or groups of that we're interested in and that are being represented in our study. And so with coevolution, the one thing people generally think of is co-speciation. And in this case, when we're looking at cophylogenies, we would see that a bee in the green would track and branch off in its phylogenetic tree identically to how the flowers were branching and speciating. But even if we have cophylogenies and we map them on top of each other and they aren't congruent, it doesn't mean that we don't have coevolution. There's actually coevolution that can occur when there are slightly different evolution trajectories by the species we're interested in. So one instance is host jumping, in which this case you could see that in green, if we were thinking of bees and flowers, a bee species was co-evolving with a certain flower species, and at one point in time, it jumps over to a new species that it starts co-evolving with. We also can have duplication events, which is where two species are arising at some point in time to evolve with the same flower in this case. Or there's also events that we refer to as missing the boat. Now, missing the boat tends to really is commonly referred to when we have an introduced species. And that means that you may find closely related flowers all near each other that have a bee specialist. But why does it not have a bee that specializes on it? And that could be because originally back in evolutionary time, there might have been a bee, but as we translocated that flower to a new location, whichever host co-evolved with it got left behind. So in other words, it missed the boat. So our project, we wanted to look at if sloths and the algae that grows in their fur and makes them look green actually co-evolved together. So to do this, we first made what's called a dated phylogenetic tree. So we used fossil calibrations from things like the ancient sloths that Emily had spoken to about before. And we had put dates on when our two major groups that we were interested in had shared a common ancestor and then started to split off from each other and create new branches on their tree. We used phylogeography, and what this allowed us to do was take our phylogenetic tree and map that over geographic locations of where the sloths were sampled from to see if any of the structures that we were seeing on this tree were a result of the actual biogeographic location that the sloths were in. And then after that, we could map our sloth and algae phylogeny on top of each other and see if there were any coevolutionary events that occurred to tightly tie the sloth and the algae. So for our sloth phylogeny, what you can see here is the map of Central and South America. And these colored lines are actually just connecting the tips of our phylogenetic tree to the areas that we had sampled to see if there was some type of geographical gradient occurring based on structure. We also dated this tree using the fossils. And so we found that the two lineages of modern day sloths speciated had shared a common ancestor about 25 to 26 million years ago, which is in line with most other molecular studies and about when the sloths arose in evolutionary time. And we also found that Bradipus torquatus, which is a three-toed sloth found only in Brazil, actually diverged much earlier on than the other modern day three-toed sloths. And it actually diverged and is an older lineage than our modern day two-toed sloths. For our algae phylogenetic tree, once again, everything is colored by sampling location. And the big difference between this tree and what we had ended up finding in our sloth tree is that there is a lot more branching going on and a lot more tips on this tree than what we were expecting to see. And this was a result of the algae in every particular sampling location had a lot of genetic variation. And so the sloths in those areas had quite a variety of genetic strains of their algae. So here we had gone to the next step of testing coevolutionary scenarios. And so in black is that sloth tree I had showed you. And in blue is the algae tree just mapped directly on top of that. And what we found when we tested for these different scenarios is that in three-toed sloths, their algae actually did directly co-speciate with those sloths. And so for Bradipus torquatus, which I mentioned, that three-toed sloth had diverged much earlier on in evolutionary time, we found that its algae actually diverged much earlier in evolutionary time as well. Interestingly, this was not the case with two-toed sloths. We'd actually found a situation in which the algae that's found on three-toed sloths host switched and jumped to two-toed sloths at some point in evolutionary time. And then once it arrived on two-toed sloths, the species of algae that are present on those two did not diversify or speciate in any way, but they did track the same evolutionary pattern of the two-toed sloths. So in conclusion, what that really told us is that there is coevolution between sloths and their algae. They have evolved through time together. And with three-toed sloths, it's actually quite close-knit, following each branch of the tree with co-speciation. But then at a later time, there was a host switch to two-toed sloths. The three-toed sloth Bradipus torquatus that's in Brazil, based on the fact that it had co-speciated with its algae and diverged much earlier on, actually has an earlier its algae is from an earlier evolutionary time than what the algae on the rest of the three-toed sloth species. And so what does this tell us? We are looking at this coevolution in these two sloths, and we kind of wanted to explore deeper because sloths are an entire ecosystem that they carry on within their fur and on top of them. Sloths are a host to not only algae, multiple species of fungus, but they have an entire diverse group of invertebrates that live on them and specialize on them. So they have moths, which are flightless and corpophagic, which means that they eat the sloth poop. And these moths rely on a sloth for every life stage, from the time that they're larvae to adult to even be able to reproduce, they need a sloth. They have ticks and lice, and the ticks and lice that are found on these sloths are species that are specialized to them. They're not found on other groups of organisms. And they also have beetles, and they have multiple species of beetles, and these beetles actually belong in different families. So it is a very diverse group of beetles living on them. And one of the beetles they have on them is actually a really rare type of beetle. Mammals usually don't have ectoparasitic beetles. There's only one case of beavers carrying an ectoparasitic beetle in North America, but we can actually find a species of ectoparasitic beetle that is living on a sloth. And sloths have mites, and even their mites are kind of interesting and a bit weird. So they have a carnivorous species of mite on them that lives actually outside their anus, and it actually crawls inside and eats intestinal nematodes and preys upon intestinal nematodes for the sloth. Sloths have fur mites, which may not seem that unusual because most mammals that are completely covered in fur have fur mites. And when you look at a sloth fur mite morphologically, it's going to look like a mammalian fur mite. But when a research group had looked at this genetically, they had actually found that sloth fur mites are more closely related to bird feather mites than they are any other type of mammalian fur mite. And so we wanted to be able to collect this diverse group of invertebrates living on a sloth. And so to do this, we actually started combing sloths out in the wild while we were sampling for other purposes. And we actually have been combing skins from museums. And when we get any invertebrates, we are identifying them morphologically first, and then we are planning to do molecular analysis on them. And one thing that really surprised us when we started this work is actually combing museum skins. We thought would be a really long shot. We really just wanted to try it on a couple and see if we get anything at all. And surprisingly, we have ended up finding mites, ticks, and beetles on these museum skins. And we have even found algae embedded still in some of the grooves of the sloth fur that we were able to collect. And so what is the point of doing all of that? Why do we care about all of these things that are living on a sloth? What benefit is it providing the sloth? And what benefit could it offer for conservation in sloths, which we know are highly affected by habitat degradation and anthropogenic changes? Well, host-specific parasites can actually offer a type of defense against novel parasites. And so if you have co-evolved with a whole host of different ectoparasites and things living on you, those can actually outcompete any type of novel parasite or pathogen that may be coming in due to anthropogenic changes or habitat degradation. And so what we're ultimately aiming to do is shift the paradigm of sloth conservation and urging for a change of not just conserving the sloth, but actually working to conserve all of the friends that the sloth has living on it as well. So at that point, I'm happy to take any questions that you have, and you can also contact me as well.
Hi, Emily. Thanks for your presentation. We have a couple questions kind of touching on your last slide. But do we know why algae favors sloths, and if the sloths benefit from the algae in any way?
We know that sloth hair is actually structured very well. It has grooves in it and gaps in order for the algae to be able to live in it. And it also, that those grooves and ridges not only catch the algae, but it allows for moisture to develop and stay there. So the way the shafts of hair on a sloth are designed to really have algae growing in it. There's been a couple different studies suggesting about what a benefit might be of having this algae. One of the key things that scientists think is that it is for camouflage. So they're up in the treetops. If your fur is green, it's going to be a lot harder to be able to see you if you are the same color as everything around you. And there also has been some tests looking at the fact that sloths are actually eating their fur, or the algae. It's found in the digesta. And the algae is very lipid rich. It has a lot of fats in it. And as folivores, sloths don't get that much nutrition from eating leaves. And so there is potential that this algae could actually be an additional nutritional source for a sloth.
Interesting. Cool. When you combed through the museum sloth skins, were they specimens that were found in the wild? Were zoo specimens excluded? And did you include captive specimens and compare?
We did both zoo specimens and also specimens that were found in the wild. And there was a difference. We actually did not really find anything on zoo specimens. And I think that has a lot to do with how sloths are often kept in, we focus on American zoos at this point, but how they're often kept in these zoos is they are often removed of a lot of parasites in order to maintain the health of an animal when they are brought into captivity. And we think that maybe there's just, from that type of treatment, they just didn't have as many parasites left on them at that point.
Cool. And then we have time for one last question. This is about the ticks that are species specific. Are there particular diseases that their ticks carry and or could pass to other sloths or species?
Yeah, their ticks do carry particular diseases, but we have not seen any real effect of more recently with the differences in habitat changes. There has started to be risks of sloths contacting things from their ticks, which they haven't before. But previously it was not thought that sloths could actually contract any of the diseases that their ticks carried. We do know that there are a couple diseases that they can pass on to humans from their ticks. And actually, interestingly, sloths are reservoirs for a lot of diseases in general. And so even without the ticks, sloths they can be actually carriers of leishmaniasis and leprosy. So even without things living on them that could possibly be harmful, they can pass a lot of things on to humans anyway.
Good to know. Thank you so much for joining us tonight, Emily. I'm going to bring back on Christina now.
All right.
Thanks.
Thanks
everyone. Thanks everyone for tuning in, and special thanks as always to Nestor, Bryson, Emily Lindsay, and Emily Fountain. Next week we'll be offline. Please stay safe and well while we're away, but we'll be back in December with 2 more episodes for the rest of the year. On December 3rd, we're going to be talking about wildcats on the West Coast. So learn more about our mountain lion and bobcat neighbors and the habitats they roam. And then on December 10th, we're going to be doing our own little virtual version of our annual holiday bazaar. We're going to have an event called Science Craft Fair, so stay tuned and join us.
And as always, if you're watching this on YouTube, be sure to subscribe to the Academy's channel. You'll get notified when Night School goes live, and some of the other live streams that we're doing. Breakfast Club is one of them. There's a great one coming up on Tuesday. But thank you so much again for tuning in. We love doing these with you. And have a good night. Have a good week. Bye.